All right, what's up, gang? How we doing? Um, Today we're going to be talking about Mendelian genetics, and this is such a kind of key piece of our human understanding about how life puts itself together, passes itself on, and changes over time. And it all starts with this little guy, Gregor Mendel, who was an Austrian monk. And he was, had a lot of free time and did monk stuff. And what do monks do? They like eat food and like pray and like clean. And he gardened. Okay. And through that gardening, he created knowledge about a key piece of how the universe worked. So this was in 1865. So, we're pushing almost 200 years ago. Um, And we didn't even know DNA existed until 1869. We didn't even know that that molecule existed. Okay. And it wasn't until 1928 that we determined that DNA was the tiny particle that passed on traits from generations, um, from parent to offspring. Okay. And Mendel figured out that there must have been some kind of tiny little particle, something that was passed between parent and offspring before we even knew that DNA existed. Okay? That's how crazy this is. And how did he do it? Well, he used this little pea plant. All right, this is obviously a pea plant. And if we zoom in all the way in here, now we know that inside of these little peas, we've got a tiny little particle that says, hey, this is going to be a green pea, or it's going to be a wrinkly pea, or it's going to be a yellow pea, or it's going to be a long pea pod, and it's going to be a tall pea plant. Right? We know this now, and we know that these little letters make it up, and the code, has three of them make a codon, which codes for an amino acid and a protein and blah bitty blue bitty blue. Okay, but he didn't know any of that. He just basically had a paintbrush and some scissors, and he did some gardening, and he figured that this is how it worked. Okay, so we're going to kind of talk about his style of genetics that he kind of discovered. But let's kind of get into this. So, what did people think before Mendel? So, before Mendel, there were some ideas. There were some incorrect assumptions about how info was passed between generations. Okay. And let's talk about these because these are some pretty interesting concepts. So, for example, we've got over here, we'll draw out a little plant, got some little leaves on it, like so, and this is a tall plant. Okay. And we take a little tall plant and a little shorty plant. What's up, shorty? We got a shorty plant. Shorty plant. Boom. Okay, we mix these together in holy matrimony. You know what I'm saying? All right, we breed them. I don't know why that's funny to me, but it is. And this is, what we do when you garden. You can specifically, bring a short plant and a tall plant together and make them mate. Okay, you can do that by cutting off the male parts of one plant, et cetera, et cetera, et cetera, et cetera. Basically, taking a little paintbrush from the other male parts and rubbing the little pollen granules on. Okay, and then we get, I didn't like the way that looked. Looked sloppy. Okay, and then let's say we get the result, and the result is we got some kind of like in between. Oh, that leaf looks a little too big. Okay, so we got a medium plant. Okay, so we went from tall and shoddy to a medium. Okay, and people thought, okay, well, this makes sense. We blend the traits together. Okay, that's what happens when you take a big one and a little one, you blend the traits together. Okay, and this was called blended blended inheritance hypothesis, hypothesis, a low idea. Okay? And this is incorrect. This has been rejected by evidence. Okay, we'll talk about how it has been rejected. Rejected by evidence. Okay, so one of my favorites now, that was one hypothesis, is this blended adherence. One of my favorites now is called this, the hypothesis of, move this keyboard closer, the hypothesis of preformation. Okay, this one's nifty. Bring that up, make it nice and big. All right, and I'm gonna bring up a little picture here. You can draw out a little simplified version of this for your notes. Okay, right there. All right, look at that. So this was the idea that inside of every sperm was a tiny little man. I really wish this one was true. Like, so there's another universe out there where this is true, and it's a better place to live. And inside of every egg was a tiny little woman. Oh, it's so good. Okay, this is obviously rejected. Rejected by evidence. Much to our consternation, we've never found a tiny little man inside of a sperm. Ugh, it's so annoying. Rejected based on evidence. Nor a tiny little woman inside of an egg. But if this were true, what it would mean is that after the sperm fertilizes the egg, after fertilization, okay, the man and lady would fight to the death for who was born. Oh man, I wish this was true. Obviously this part, I don't know if this is what they really believe, but that's what it means to me if you think about it. There's only one way this issue could resolve itself, and that's with a knife fight in the embryo. Okay, so this is obviously also rejected. It's not, unfortunately. As much as I want it to be true, it's not, and that's the beauty of science. We got to reject or support our hypotheses, and unfortunately, that one's rejected. I know. Cry me a river. Okay, write about it in your journal. Okay, so then, how then are traits actually passed from generation to generation? How is it that this works? And let's kind of take a little look at some terms here, and then we'll get into this. All right, so we'll look at first kind of some chromosomes and stuff like that. All right, so let's look at this. So over here, let's see what this looks like. Get rid of that. All right, we've got 23 chromosomes and 2 copies of each. All right, so we're going to draw chromosome 1 out here so we can get an idea of this. That's chromosome 1. Chromosome 1. And these are where our DNA is contained. We got 23 of them. They're like little lines. And in there is all just DNA. Okay? Version A. We got 2 versions of every one. Okay? And then we've got chromosome, oops, caps lock. Chromosome 1 version B. Okay? And so that means we got 2 copies of every gene. We got version A and we got version B. Okay? So like right here, we've got a gene that has to do with height, let's say. All right? So this one, let's say, is a big T gene and this one is a little t gene. Okay? So this would be, I'm sure you guys have heard some of this before. I'll make these a little larger. Okay, this would be for like tall. Okay. And this would be for short. Okay. And this Since it's a large T, that's how we represent dominant. Okay, and that doesn't mean better. It just means it's the gene that's expressed. Okay, and we'll kind of define that, I think, in a little bit. We'll put it here. It doesn't mean better. We'll put it here. It does not mean better. It just means expressed. It's the gene that's on. Gene that is on. Okay? That's what dominant means. Okay, this little t, that's how we represent recessive. Okay, and it doesn't mean that it's not the most prevalent. Like, you could have a recessive gene that is super prevalent. Okay, for example, having 5 fingers is a recessive gene, but it is super prevalent. Right? Most people have 5 fingers, but that is a recessive gene. Okay, O blood type is a recessive gene, and it is the most common blood type. Okay, so dominant doesn't mean the most popular or better or anything like that. It just means it should be called on or always on. That's what it should be called, always on. Recessive is only on if other one's not there. Okay, so this is only on if dominant is not present. That's really what it means. Okay, it's about gene expression. Okay. Yeah, I actually put this in better language in these notes and I forgot to do it. Okay, but that's fine. It's cool. It's cool. I got it. All right, so let's kind of look at these. So these are our chromosome versions. Should put that on there a bit ago, but I forgot. And let's kind of look at this. Okay, I got a little picture here to kind of take a gander at. All right, so this is, well, a little low res, but that's okay. So these are chromosome 1, 2, 3, 4, 5, 22, and then our sex chromosome. So we've got 23 different types of chromosomes, and we have 2 versions of each. All right, so that's what these are. That's version 1, and that's version 2. That's version 1, that's version 2, that's version 1, that's version 2. Okay, so that's what we're talking about. All right, so now let's get into defining some genetic terms. Okay. So let's just define some genetic terms so we can all be on the same page with this nomenclature. All right, so we've got over here, we've got a genotype. Okay, what is that? Genotype. And this is the genes that are present. Okay, so if we look at an organism, that's the genotype. If we look at the genes inside of it, all right, that's the genotype. So for example, if we have, remember we have 2 versions of every gene. So for example, if the person or the plant or whatever has big T, that's its genotype. Okay, and if it's big T, this is called homozygous dominant. Homo meaning same, and the zygous meaning zygote. Okay, dominant. So it's the same. It's 2 dominant, so it's homozygous dominant. Okay, and then over here we have the phenotype. Pheno means like what is seen, what is made, what is manifest, like phenomenon, what is manifest. Okay, so in this case, the plant will be tall. That's the phenotype. Okay. All right, we could have heterozygous, which is big T, little t. Hetero meaning different. Heterozygous, meaning hetero means different. Different zygotes. Okay. And this plant will, what would its phenotype be? Remember, this dominant one is on, and the dominant one is tall. So this plant shall be also tall. Beautiful. Different purple though. Let me do the same. Purple. Blue and yellow, purple pill. Okay. All right. Last one, but not the leasty. I'm doing these different colors. I had to change my colors because, I don't know, people couldn't read them. Anyway. I'm a little sad about it. I lost my like neon blue. All right, so there we go. We got homozygous, and this one is going to be recessive. Homozygous, recessive, okay? Which means we've got two recessive traits. Here we got one of each, heterozygous. Here we got two of the same dominant. So it's homozygous dominant, homozygous recessive. And what is this plant going to be? A shoddy. It's going to be short. Cool. So, genotype, phenotype. Phenotype is what we see. Phenomenon. It's what we see. Lightning is a phenomenon. Okay. Genotype. It's the genes that are present. Easy peasy. Okay. Science is so fucking easy. Don't ever get it twisted. It's the easiest crap on the planet. Okay, so let's define these, write these out. Genotype is the genes we have present. Okay. And another way we could define this, it's the type of alleles, which we'll define in a second. Alleles. Is it like this? Alleles. How do I spell that word? That's what I did the first time. Stupid. Types of alleles. Okay, and we'll define what those are in a second. Okay, so genotype is the genes we have present. Phenotype. Phenotype is the physical characteristics that we see. Okay, it's what is manifest, what is actually the physical trait. Okay, that's phenotype. All right, allele, two L's in the front. Allele. This is the different versions of the same gene. Okay, so like big T is an allele and little t is an allele. Okay, and you can have, we'll talk about at the end, you have multi-allelic traits where you have like 3 different copies. A lot of them only have 2, dominant, recessive. Okay, but some have 2 dominant, some have 2 that are like co-dominant, some have like multiple different genes that we could have. We could slot in like 3 or more. Okay. And then the last thing that we're going to kind of mention is called trait. Okay, so the trait is the general characteristic that we're talking about. So for example, in that case that we've been talking about, this is the general characteristic, okay, affected by the genes. Okay, so for example, we've been talking about height. Okay, that's the trait. Okay, so genotype, phenotype, allele, and trait. Know those. All right, cool. All right, so now what we're going to do is what Mendel did. We're going to cross 2 homozygous pea plants. We're going to cross them up. All right, so let's kind of get our little pea going here. Let's really lay our pea on here. Draw a nice, it's a nice tall boy and a little shoddy. Okay, easy. So these would be our parents. It's mommy and daddy. Okay, and this one over here is gonna be, this is homozygous, right? Homozygous. So if this one's tall, what is its genotype, its genotype is going to be a big T, a big T. Okay, so it is homozygous dominant, dominante. Okay, this one is short. It's a shoddy, so that means it's got to be homozygous recessive. It's the only genotype it could be. Homozygous recessive. Cool. Easy. All right. Now we're going to cross these. We're going to breed them together. So we're going to take a little paintbrush. We're going to rub it on the little anthers of one of them. Okay. And then we're going to paint it on the other one. And then it's going to make peas, and we're going to plant those and grow them up. And what are we going to get? We know. If we know these are homozygous, we know what kind of plants we're going to get. And if you can't think about it, can't guess it, we'll show you. So we're going to cross these together. Breed them. Hey, I'll be right back. I'm going to go outside in the shed and breed the peas. It's a fun thing you could say as a gardener. Okay. And what are we going to get? Well, you're going to get all tall plants. Okay, and we'll break that down as to why that is. But their genotype is going to be this. It's going to be heterozygous. Okay? Heterozygous. Heterozygous. And we'll break this out with a Punnett square here in a minute. So our genotype is heterozygous, or we could say big T, little t. And our phenotype is what? All the kids. Phenotype is tall. Okay, all the offspring are going to be this. We'll put that up there. All offspring will be this. And I succeeded. Okay, good. All right, and this is called the F1 generation. This is called the F1 generation. It's not Formula One. It stands for filial, which means child. So that F stands for filial, means child. Okay, so the first child generation. All right, now let's cross two heterozygotes, and we'll see what we get. Okay, this one's a little less interesting to do the cross, so we'll do the cross now. All right, so now let's cross, which just means breed, two heterozygous and do a Punnett square and kind of see what's going on. People love these Punnett squares. I think that's how you spell it. I don't know why it's mad at me. People love these. People get excited about these. People will riot for Punnett squares in a biology class. People will flip over cars and start making Molotovs. Just be like, when are we getting to the Punnett squares? Are we going to do Punnett squares in this class, Tori? Tori, can we do the Punnett squares again? That's why I saved them to the end. Because if I did these, it's just, in the beginning of the class, it'd just be like, why aren't we doing the Punnett squares? I missed the Punnett squares. I don't know what it is. I'm going to be honest with you guys. I think they're kind of boring. But that's probably just because I don't know. I just hate rectangles. Or I'm more of a rectangle guy, I think. Anyway. All right. So this would be the F2 generation. Filial 2. All right. These are the kids of our kids. Okay. And you can inbreed plants. It's okay. Don't do that. Don't do that with humans. It's bad. All right, mess the offspring all up. Okay, so we got basically what we got to do. You know, when we were talking about meiosis, right? You're having your genetic material, so you're going to make gametes. You're going to make sperm and eggs, and we want them to be half of the genetic material right, in the egg, because then when we take the sperm and mix it together, we want to make it whole again. Okay, so to make a sperm and an egg, they only have one version of each chromosome. Okay, so like a sperm is only going to get version A, or it's going to get version B. So then when you mix it with the egg, you get version A and B back together again. Okay, so like in a sperm or an egg, it's like we delete that and we delete that and then we delete that and so on and so forth. We just have now one copy in a sperm or egg. And then when they combine, that's called fertilization. Then they have the two copies again. Okay, so each one of these represents, like let's say this is mom up here on top. Each one of these represents an egg. And each one of these represents a sperm mutation. Okay, and this first one, let me put this in here. This egg, let's say, got a big T. Okay, and this sperm got a big T. Okay, and this egg got a little t. Oops, I want that in this kind of nice green color. Okay, this sperm got a little t. And this sperm got a little t. All right, then each one of these represents a fertilized, okay, this represents a fertilized, now embryo. We'll just call these different embryos. Embryos or zygotes, if you remember, that's what they're called. Okay, each one of these are basically what the kids would be, because right, we combine the sperm with the egg and then we get the kid. Okay, so to do this, all we do, it's so easy. This is why people like it, because it's kind of fun. Okay, we go like this, boom, we take that and we take this and we put them together. And what are we going to get? We're going to get T. Let's go copy it and bring it down. T right there, and then T. So that's one possible baby we could get. Okay, then we'll do this baby. We'll do this T, bring it over, bloop, and this T, bring it down. And we always do the dominant first, just for like sake of clarity. Okay, makes it easier to read. Okay, then this, we'll do this one, which is we combine this egg with this sperm. These are just the different possibilities we could get for these kids. And we'll do this one. Bloop, like so. Okay. Then we bring this one over here, this little t and this little t. Okay, bring that on down. And bring this on over. Boom. So these are our possibilities for our genotype. Okay, so this is our F2 generation, crossing two heterozygotes. And we got these possible genotypes. Okay, these are called the genotype ratios. The possible genotypes, we can get the frequency of them. How often do they show up? I'll always be doing that. Okay, genotype ratios. So we've got one big T, big T. We got one big T for every two heterozygotes. For every two heterozygotes. Doot, doot. And let me actually kind of highlight those in that color that I just did. So we'll make these like so. Get rid of that. Make that big. Okay, so that one's that blue. And then we got this purple, right? Like that. And then we got one homozygous recessive. Okay, we'll do that in, I'll just do it in gray. For every one little t, little t. Okay, so it's a 1 to 2 to 1 ratio if we cross heterozygotes. All right, but like that. Okay, and then, but then we also have a phenotype ratio. Okay, which is going to be what? This plant here is going to be, we'll do it in green. It's going to be tall. These plants would be tall. Oops, I want it in green. Mine's going too fast for my fingers. Okay, this one's also going to be tall, right? Because it's got a dominant. This is going to be tall because it's got a dominant. And this is going to be short. Okay, so this is 3 tall, 3 to 1, right? 3 tall for every 1 shoddy. Easy. Science is so easy, don't let anybody tell you otherwise. They're lying. Okay, so that's what happens when we cross 2 heterozygotes. Okay, so we started with homozygotes. When you do that homozygotes, you're just going to get heterozygotes, and then we can cross those, and we're going to get these ratios every time. A 1 to 2 to 1 for the genotype ratios. You're going to get, the same amount of homozygous, dominant and recessive, 1 to 1 ratio. And then you're going to get, for every one of those, you're going to get 2 heterozygotes. And then for the phenotype, you're going to get mostly tall and 25% short. 75% tall, 25% short. That's what you can think about these as percentages too. So this would be like 25, 25, 50. And this would be 75, 25. Okay. All right. So let's just get a little practice with this. We're going to do a little guessing game. Okay. So we're going to do a little guessing. Possible genotypes. Okay. And I'm going to put a little info up here and I'll show you a picture. Okay. So the gene, we'll talk about something else. We'll talk about rabbits right now. The gene for black rabbit fur is dominant, okay? And that would be a big B. Okay, the gene for recessive the gene for white rabbit fur is recessive. Okay, and that would be little b. Okay, and so I want to bring up this picture here. Okay, so what about this? Okay, so what do we see here? The phenotype here for this is obviously, what? Guess for me for the first one. Phenotype. For this little guy, it's obviously white. It's white and snuggly. I'm white and snuggly. Don't catch me white and snuggly. Don't catch me white and snuggly. And I'll stop with that. I can keep going, but I'll stop. I'm white and snuggly. Okay, so the genotype for this would be what? Don't mess it up. It's going to be little b, little b. Okay, right? Little b, little b. That's the only genotype it could be. Okay, and this one is black and snuggly, obviously. Look at them. Look at them. I want to get them. Okay. What is the possible genotype for this one? Give you a second. There's a secret message in there. All right. Possible genotype. It's kind of a tricky one. It could actually be homozygous dominant or heterozygous, right? It could be either of those. Okay. Now, a little tricky one, a little tricky trickster. What about a rabbit with regurgitated Italian food on its hoodie? What about that? What would the genotype of that rabbit be? That would be B rabbit. Duh. Okay, we'll end this one here.